Hi guys, welcome to this Sunday session. Sorry I'm a little bit late, just having some technical difficulties with my screen. It every time I touch the wire that plugs in the screen shuts off for some reason. So I'm gonna try and not move anything during this lesson, and hopefully it all works. Just give me a quick thumbs up or something if you can hear me okay. Just want to check everything's up and running. Perfect. Hi Jess, hi Zoe, hi Clara, hi Esme, hi Laura. Can't do it to everyone, but nice. How is everybody this evening? What have you guys been up to this weekend? Anxious for the lesson? Shouldn't be anxious for this. Although, I guess probably starting to get a bit of exam anxiety or nervousness building probably as we enter the final few months. You've had mocks the last 2 weeks. Yeah, I think lots of people have been doing mocks the last few weeks. How have they gone? Or I think some people still are doing mocks like just before half term or just after half term. So yeah, it's a busy revision period for lots of people. Yeah, then this week. Okay, yeah, nice. Cool. Well, I guess it's already a few minutes afterwards, a few minutes after 5. So let's just dive straight in with our lesson. So just remember, I haven't done a YouTube session for like a few weeks. Well, I think it's a few months even. So I'm trying to remember how to do it. It's a little bit different to our normal classes. So I think that should be up and running. Are you guys now looking? Yeah, you can see a PowerPoint, right? Good, Nesta. Well done. Yeah, that's a very good sign. Tips on how to paraphrase. Yeah, we could maybe, if we've got time at the end, we can talk about that. For essay questions, are you an AQA student then? I presume, because I think it's just AQA that do long answer essays. Other subjects you do like 6, 7 markers, but not full. AQA do a 25 mark essay, but it's not. If you come from like an essay subject background, it's not really an essay. It's just stating facts. You don't really have to write a conclusion or like an introduction. They call them essays, but they're not quite. But anyway, yeah. For the rest of the time, unless it says you need to write in continuous prose, I would recommend using bullet points. I know some teachers don't like you to do that, but from the examiner's point of view, they like bullet points. It's easy for them to mark. It's easy for them to see where to award the marking points, and it actually, I would say, it gives you a better chance of like getting the marks. If it's all buried in a big block of text and the examiners are marking the paper very quickly, which they have to do, there's quite a big time pressure on them. Yeah, make it as easy as possible for them to give you the marks, and it's got to be a good thing. So yeah, today's session, we're doing respiration and glycolysis. So I know this is a topic that a lot of people find difficult. It's usually when you do it at the start of year 13. So I thought it'd be good to recap it today, because probably you guys haven't looked at this for a little while. Yeah, it's pretty nice we don't have coursework for biology. Although, I don't know, some people like coursework. Coursework, you can, at least some of your grade already. Like if you get a good mark in your coursework, it kind of takes the pressure off the exams a little bit. So in some ways, coursework is nice. But yeah, some people prefer exam, some people prefer coursework. So yeah, just before we start our session today, just to give you a little bit of info on these exam crash courses that we're going to be running at Easter. So some of you guys might have heard of these, or actually probably not, because we didn't run them last year. And then the year before that, most of you guys, I presume, wouldn't have been doing A level. So you probably haven't heard of them. So basically, there are these exam prep courses we do over the Easter half term. We do them in biology, but also the other 3 subjects: physics, chemistry, and maths. So you can go to all of them if you're super keen. And in fact, there's actually a discount if you do go to all of them. So yeah, these are really, really helpful short 2-day intense courses, and we go through a few different things. Like a typical day, we'll start with some exam technique. So this is going through command words, analyzing questions, how to score marks, and how to jump through the right hoops to let the examiners give you the marks, and sort of tips to make sure that you apply the knowledge in the best way to do well, essentially. So yeah, we start with exam technique, and then we go through some of the more challenging topics. So actually respiration would be one of the topics that we would usually go through, and photosynthesis, because people don't like that one. Maybe do some of the nervous system stuff, meiosis. In the AS, I know that lots of people don't like plant transport or the cardiac cycle and the heart, so we would do those topics. Basically all the ones that people typically find very difficult and tend not to do so well on. So we'll target those. Then we also give you this booklet. So this is a really, really useful learning resource. You'll be writing stuff in it during the day, kind of taking notes in the booklet, but then you also take it away, and there's a bunch of useful stuff that you can look through that in your own time, like extra questions to go through, sort of tips, sort of summarize, condensed notes, sort of, I don't know, cheat sheets if you like for the different topics. Yeah, so really helpful. And then the final thing, during the day, we're going to run these as seminars. So it's a little bit like a university lecture. So there's a group of people in the room and a lecturer. So you can ask questions during the session, but I know some people don't like to necessarily ask a question from a big group, and fair enough. Like, I don't like doing that either. So what we have at the same time is, if you've got a question during the lesson or afterwards, we've got this messenger service. So we've got other tutors on standby. So if you've got a question, you just WhatsApp the tutor directly to a teacher, and they'll reply straight away. So you get sort of live answers on anything that you want extra help on. So yeah, another really good feature. And yeah, I'll give you a little bit more information on these at the end, but essentially they're in all of our subjects. They're run out of Regent's University in London, so that's like bang in the middle of Regent's Park, really nice location. Or you can watch the live stream. So we're going to record them and stream them a bit like we're doing now. Yeah, Mohammed, good question. So they're both in person or online. So you can do either. If you live in London, probably better to go in person. Well, up to you really. If you live further away, probably the live stream would be better. Cool. They're for all exam boards. So yeah, any exam board, this will be relevant for you. We focus on the stuff that is sort of the same between all of them. Any bits that are exam board specific, we'll tell you. So yeah, I guess we should probably make a start. If you've got any questions on that, I'll hang around at the end and we can talk through that in more detail. But yeah, let's get on to our main lesson for today. So we're looking at aerobic respiration. We're looking at the process of glycolysis. So this is the first step in respiration. And we're going to look, if we have time, at anaerobic respiration as well. The seminars are for both AS and A2. So we run, actually, sorry, I said it was a 2-day seminar. That's not true. It's 2 separate days. One is for AS and one is for A2. So if you do AS, you can only come to the first one, really, because we're going to be doing the AS stuff. I think that's on the 13th. And then the 14th, that's the A2 one. So if you are an A2 student and you're sitting exams this summer, you can go to both days. They're both going to be useful. You can revise the AS on the first day and then do the A2 stuff on the second day. They are, I think it's £99 for the in-person and £79 for the online one. And if you like I said, if you go to more than one subject, you get discounts. So I think it's like buy 3 get 1 free, something like that. So if you do all 4 of those subjects, or you want to split them with a friend, you can get quite a big discount by doing multiple courses. So worth considering. Cool. So yeah, respiration's in all the specs. So these are the spec points. I won't really talk about them now, but you should all have a copy of your specification anyway. But yeah, everybody does this because it's a really crucial part of biology. So yeah, let's recap a little bit about ATP and what we sort of need energy for in the cell or in any organism. So yeah, you can tell me what is ATP firstly. And I guess as a follow-up question, what do we need ATP for in the cell? Yeah, so good answers, yeah. So it's an energy store. So we say short term. And it's like very short term. ATP typically only lasts a few seconds before it's used. So like really short term energy store. Yeah, it's the universal energy currency. I like that. In cells, or you could say in living things altogether. It is a phosphorylated nucleotide. Very good. And we actually make it by combining, don't know if anyone said the equation yet, possibly, or maybe not. So we combine ADP and PI. You'll often see it written like this. This stands for inorganic phosphate. This can be turned into ATP plus H2O. People always forget to put the water on in the equation. It is actually a condensation reaction to do this. So, well, actually, I should probably draw this a little bit differently because it's a reversible reaction. We can draw it like this. So this way is ATP synthesis. This uses ATP synthase, the enzyme. That's not how you spell synth. Okay. And then the reverse way, the hydrolysis reaction, this uses ATP hydrolase, or sometimes just called ATPase for short. Yeah, these sessions are recorded, so they just get uploaded to YouTube straight after. So yeah, you can look back at them. What is it in inorganic ion? Yes, good question, Josh. So anything that's inorganic, or I should probably say what's organic, that'll be easier. So anything that's an organic molecule has carbon and hydrogen in it. So most of the things we talk about in the biochemistry module, like carbohydrates, proteins, lipids, they're all organic molecules. They contain carbon and hydrogen. So when the phosphate is attached to the ADP, this is a nucleotide. It's an organic molecule. We call it an organic phosphate. When it's by itself, it's inorganic. So yeah, that's why it looks like that. Cool. Yeah. So then some of you guys have started saying what we need ATP for. So there's like hundreds of processes in the cell. It's used to power lots of different things. So some of the ones that you've got already put, yeah, active transport. Nice. Mitosis, good. Muscle contraction, yeah, like that one. That's actually a very big sort of sink for ATP. We use a lot of ATP for muscle contraction, particularly if you're like very active. Yeah, endo and exocytosis. Nice. Nerve impulses, yeah, good. I guess you could say like specifically, that would be the sodium potassium pump. So you could say like, I guess it's kind of like active transport. Yeah, Dan, yeah, it is right. You shouldn't really, this one wouldn't be a good answer. And in fact, yeah, and I put it first as well. So, yeah, better to say, yeah, immediate would probably be a better answer. Cool. So anything else? Protein synthesis? Yeah, that's another good one. A lot of ATP used up in that. Nice. And yeah, and lots of other good examples that you guys have come up with. I guess we should draw one out, unless it's on the next page. No, I don't think it is. So yeah, we've got a yeah, basically an adenine base. Bonded to ribose sugar. In fact, maybe I should draw it. Oops. It's zoomed in for some reason. So ribose sugar, adenine base, say purine. So it's got a double ring structure. And then we've got our 3 phosphates like that. So when we turn it to ADP, we lose this last phosphate. You also get AMP, so adenosine monophosphate. That's when it loses 2, but we don't really talk about that at A level, but it is possible. So it can actually like lose another phosphate. Yeah, that's a good question. You do get GTP, and you get, I actually don't know if you get UTP, but you definitely get GTP. And yeah, I haven't really heard of CTP actually either, but GTP is used quite often in metabolic reactions. I presume that is a CTP. Don't know about UTP. But yeah, there are other ones. This is just the one that is used the most. So that's why we learn about it. Cool. So that's ATP. Let's look at how we make it. So this is kind of an overview of the whole process of respiration. So yeah, we're only going to look at the anaerobic pathway today, but preferentially the body would go down the aerobic pathway to make ATP. This is because if you do aerobic respiration, you make a lot more ATP per molecule of glucose. So you get around, it's between, like, you get different answers from different specs, but between roughly 34 to 38 ATP per molecule of glucose if you do aerobic. Whereas if you do anaerobic, you actually only get 2 ATP. So yeah, the yield is much higher for aerobic. And the other benefit of aerobic is you actually don't produce any really nasty chemicals. So all you produce at the end is water and CO2. So yeah, it's both of those are not particularly problematic. The water just gets sort of mixed in with the rest of the cytoplasm, and the CO2, it can be bad if it builds up too much, but we've got a pretty good mechanism for removing CO2 from our body. It just travels into the bloodstream, and then we breathe it out in our lungs, so we can get rid of that quite easily. Anaerobic, on the other hand, this produces either, if you're like a mammal, we make lactate or lactic acid, or if you're something like a yeast or a plant, you would make ethanol and CO2. Both of these products, lactate and ethanol, are more toxic than CO2. So when they build up too much, this is a problem. And you'll have come across lactate buildup. We get this when we do like intense exercise for a long period of time. You're going to do a lot of anaerobic respiration. you get the lactate building up, and you can feel the sort of burning sensation in your muscles. So it's quite a nasty product. You can't make too much of it without causing problems. Yeast have the same problem. They make ethanol. We use this to our advantage. We use this to make alcoholic drinks. We use this to make bread rise. So it's a useful reaction for us. But the yeast, they make the ethanol, and eventually, if they're making it, so if you've got a fermentation going, you're making some wine, for example, eventually there's so much alcohol that the yeast are killed off by the alcohol, like the waste product. So yeah, it ultimately, they can't do it for a long time. Someone's saying something about, lactate is reoxidized to pyruvate. Yes, exactly. So if it was reoxidized to pyruvate, you would just go into the link reaction. Spot on. So we call that repaying the oxygen debt. It takes a little while. After you finish exercising, you've got to sort of reoxidize all that lactic acid. So that's why you keep breathing heavily for quite a long time after you've finished doing intense exercise, because you need oxygen for that process. So yeah, the lactic acid is not wasted; it's reused. Yes, this is A2 content. If you're year 12, this might be a little bit difficult to follow along, but yeah, you could, like, hopefully you'll get something out of it still if you haven't seen this before. So yeah, let's look at glycolysis in a bit more detail. So whether you're doing anaerobic or aerobic respiration, you always start with glycolysis. So it's a really important process, and we go from glucose into pyruvate. So let's look at how that actually happens. Just like very quickly, or just to maybe throw it out there, what is the equation for aerobic respiration? This is something you should have learned at GCSE, and if you haven't, definitely learn it for A level. And as a little bonus, is that you get 2 equations for the price of 1. Because if you know the equation for respiration, you also know the equation for photosynthesis, which you do also need to know. So it's just the reverse. So if you have one, you know them both. So that's quite nice. So you can give me the word equation, because it's probably easier to type if you want, or you can give me the full chemical equation. Yeah, nice, Eleanor. And you've balanced it. Perfect. So we've got, whoops. Six O2 goes to 6 CO2 plus 6 H2O. Yeah, exactly. So glucose plus oxygen gives us carbon dioxide and water. Nice. And always just remember 6, 6, 6, really easy, and 1 glucose. And then you've got it balanced. And obviously for photosynthesis, you literally just reverse it and it would go in that direction. So yeah, this is a mitochondria. It's not really involved in glycolysis or anaerobic respiration, because glycolysis takes place in the cytoplasm. If you're going to go down the aerobic pathway, then the mitochondria is involved. So there's 2 different places stuff occurs. You've got the link reaction and the Krebs cycle. They occur in the matrix, which is sort of the cytoplasm of the mitochondria. And the oxidative phosphorylation, or electron transport chain, happens on the inner membrane. So there are lots of proteins embedded in that. So this is really important for electron transport chain. And this one is link plus Krebs. Respiration and photosynthesis can happen at the same time in a plant. Yeah, definitely. They would be happening in different places though. The respiration would be happening in the mitochondria, and the photosynthesis would be happening in the chloroplast. But the processes could be happening simultaneously. Yeah. So how are you guys feeling with that so far? What are you saying, Florence? Oh yeah, cytosol. Yeah. So cytoplasm is everything, including the organelle. Cytosol is technically just the actual like liquid, sort of the plasma. And then what was the other question? What's the difference between glycolysis and glycogenolysis? So glycolysis is the breakdown of glycogen. And then, sorry, no, glycogenolysis is the breakdown of glycogen into glucose, which is used as blood sugar. And glycolysis is the breakdown of glucose into pyruvate, which is the start of respiration. So glycolysis is to make ATP. Glycogenolysis is to increase blood sugar levels. So they're kind of, yeah, they would be happening at different times. So let's do some questions just quickly. Check how you guys are getting on. So first question, in respiration, where does glycolysis take place? For 1 mark. Nice. Just in the cytoplasm. And where does the electron transfer chain take place? Yeah, in the inner mitochondrial membrane. Yeah. Yeah, we can call those the cristae. Nice. So that's just another name for the folds in the inner mitochondrial membrane. And yeah, both should be accepted in the mark scheme. I would be a bit careful just saying inner membrane. If they haven't mentioned the word mitochondria, then I would probably specify that it's the inner mitochondrial membrane. If they said where in the mitochondria does it occur, then you can say inner membrane, that's fine. But if they haven't mentioned it, it would probably be better to say inner mitochondrial membrane or cristae. Nice. So we now got a 2 mark question, still relatively straightforward. So mitochondria in the muscle cells have more cristae, more of these folds, than mitochondria in skin cells. Explain the advantage of mitochondria in the muscle cells having more cristae. Glycolysis happens in the cell cytoplasm, not in the mitochondria cytoplasm. The mitochondrial cytoplasm we call the matrix, and that's the link reaction and Krebs cycle. So yeah, just the cell cytoplasm. Link reaction is in the matrix, yeah. So yeah, it's got more surface area. Can't write and talk. More, I'm going to put SA for surface area. For electron transport chain. You shouldn't write like this. I'm just writing it briefly for this lesson, but you should obviously write these out properly. So more surface area for the electron transport chain. Therefore, for the second mark, has anybody said yeah, more, yeah, nice, Khadija. So therefore, it can make more ATP. Or like ATP can be produced faster. Actually, I think more is fine. More ATP produced. The screen isn't clear. Is it clear for everyone else? Hopefully it's uploading okay. It might just be, yeah. Sometimes it just, YouTube will switch to like a lower rate if your network's like a little bit slow. So maybe that's the problem. So hopefully it will get better. Greater surface area would not necessarily be equal to lower surface area to volume ratio, probably more like the other way around. just increasing surface area might not necessarily increase the surface area to volume ratio, but you would generally expect it to. Yeah. So okay, let's look at glycolysis in a bit more detail. So glycolysis involves the splitting of a hexose sugar, i.e. glucose, into two smaller sugars called pyruvate. So these are 3-carbon sugars, and it takes place in the cytoplasm, as we said. So let's just break down this word, hexose. So hex means 6, and in this case it means 6 carbons. Just like a hexagon, it's got 6 sides. And O's, if you ever see OSE, this always tells us that it's a sugar. So just like ASE equals enzyme, you can kind of something about that molecule if it ends in ASE, and if it ends in OSE, it's a sugar. So just sort of bear that in mind. Yeah, and glucose is the most common hexose sugar. So it looks a little bit like this. You might, I think no spec actually demands that you need to be able to draw out glucose from scratch. You need to be able to draw like a hexagon and with the OHs. So you need to be able to draw basically just that. And it might be worth like learning where the other OH groups are and the hydrogens, but I don't think it's actually necessary. But you might find it easier for some questions. But if you are able to draw one out, you also need to be able to number the carbons. So we go from the oxygen, and then we go around the circle, numbering them like that. So 3, 4, 5, and then carbon 6 is the one that sticks up out of the ring, up like that. So technically, this would be alpha glucose, because the carbons on the 1 and 4 are both orientated down. Ribose is a pentose sugar. Yeah, it's got 5 carbons. Amylose is a polysaccharide of glucose. Yeah, nice. So a lot of these alpha glucoses joined together would give you amylose. They're all joined by 1,4 glycosidic bonds. That's an amylose. So this is a 3-carbon sugar. And what should we say? It can either enter anaerobic or aerobic pathway. So yeah, they both start with this reaction. Cool. So this is glycolysis, and let's break it down into 4 main steps. It's actually not overly complicated. In real life, this reaction does have a few more steps in it, but we only focus on the most important ones. But the actual process of glycolysis is about 10 stages to it. But a lot of them, not really much happens. So this is just the most important ones. So the first step, we turn glucose into hexose bisphosphate. There should be an S in here. It's a bit of a typo. Oh yeah, like it's written here with an S, bisphosphate. So that name, we can kind of break that down. Hexose bisphosphate. Bis or bi meaning 2, and phosphate meaning phosphates. So our glucose has been phosphorylated. We've stuck some phosphates on it. And this is kind of the weird step of glycolysis, because we actually use ATP to do that. So to phosphorylate glucose, 2 ATP hydrolyze to ADP. And the phosphates, they shouldn't actually be in this equation because they remain stuck to the glucose. Yeah, good question. Why does pyruvate not end in O's? So it's not always the case, but if you see something that does end in O's, then you know it's a sugar. But not all sugars end in OSE, just like not all enzymes end in ASE. But if it does end in that, then you know it is that. Cool. So, yeah, this step is done. It seems a bit weird because you're putting in ATP, even though we're trying to make ATP. But it's a bit like you've got to spend money to make money, that sort of idea. We need to do this first step, even though it's actually using up ATP, but then we can make more ATP at the end, so it's worth it. I guess it's a bit like buying your house in Monopoly. It costs you money initially to buy your house and put it on your piece, but if someone lands on it, you're going to get way more money, so it's worth it. So we've put in 2 ATP. We've made hexose bisphosphate. Some specs, you just call this phosphorylated glucose. So if you've seen that word before, then you can just use that instead. That's fine. Aspartic acid, yeah, exactly. It's one of the 20 amino acids. I can't remember if it's an essential one or a non-essential one, but yeah, it is one of the 20. So yeah, this hexose bisphosphate, this is unstable. The reason we phosphorylated it was to make it unstable. So it pretty much straight away splits into 2 3-carbon molecules, and we call these triose phosphate. Which is commonly abbreviated into 2 TP molecules. 3 carbons. Yeah, at least for AQA, they're releasing a statement tomorrow on what's going to be on the exam. So yeah, you should definitely look at the website and see what they actually announce, because it's going to be kind of interesting. I'm not sure if it's all exam boards that they're announcing tomorrow, but definitely the AQA one they're doing. So they're basically going to tell you whether they're going to take some stuff off the syllabus or not. I don't know if they will. We'll see. We'll see tomorrow. So that will be quite important to tailor your revision. If they're going to take some topics off. Not sure what time they're going to release it, but they just said it was going to be out on the 7th. So yeah, at some point. So yeah, next up. So now we've got 2 TPs, and we're going to oxidize our TP. So we should probably recap what oxidation is here. And you guys have probably, or hopefully, heard of oil rig before. So oxidation, I'm not going to write it all out, but is loss. Reduction is gain. So this is of, where should I write it? Of electrons plus hydrogen. So always refer back to oil rig whenever you're in doubt. Is it being oxidized? Is it being reduced? Just look at, is it losing electrons and hydrogen, or is it gaining electrons and hydrogen? So in this case, we're not really focusing on the hydrogen, so you can't actually tell that much. But other times, like in the Krebs cycle, you can see exactly what's going on. So triose phosphate is oxidized. Redox reactions always sort of occur. They're always coupled. So if something's oxidized, something else needs to be reduced. So TP is oxidized. NAD is reduced. So oxidation of triose phosphate produces 2 molecules of reduced NAD. So you might write this as reduced NAD. You might write it, and I would recommend writing it like that. Some people might write it as NADH. This is usually okay, but you might not get the mark in an exam, because sometimes they're picky. It should actually be, if you're writing like this, it should technically be written, that's the full way of writing it. They usually allow just NADH, but why take the risk? If you just write reduced NAD, you're definitely going to get the mark. So that's the safe option. The purpose of this then goes on to make ATP, ATP in the Krebs cycle. Oh no, sorry, in the electron transport chain. So this is a coenzyme. Let's put a little note on that. Coenzyme. And its main job is to transport the electrons and hydrogen, E minus plus H plus, to the inner mitochondrial membrane for the electron transport chain. So let me just, haven't really looked at the comments for a little minute. Let me just check. Cool. Perfect. So then the final step, this is the one that actually, the most important one, arguably, because this is the one that actually makes ATP. So at the same time we're reducing the NAD, that releases some energy, and that energy is used to stick together ADP and inorganic phosphate into ATP. So production of TP, this should say, no, it should say production of ATP. So of TP provides energy to synthesize 2 times ATP. So each TP, 1 TP equals 2 ATP. And we've got 2 TPs, so we make 4 overall. Florence, it's a good question. It's actually, we don't need to go into details because the math doesn't quite add up, because we put 2 phosphates in and technically we're getting 4 out. So it's kind of a mixture. 2 of the phosphates come directly from the TP because they're like located on it. We get 2 more just from the sort of in the cytoplasm, but you're not expected to know anything about that. So yeah, don't worry too much about that. I think they're going to announce, I'm not sure if it's for other subjects on the same day, but potentially they're going to announce all, yeah. Check on your whatever spec you're doing, check on their website, and they've released a statement on that subject page. So just go have a look at your subjects and see what they're saying. And they should be able to help you with that. So yeah, let's look at what we've produced so far. So ATPs, we made 4, but used 2. So the net ATP is 2. Yeah, net gain. Net is an important word. This is like how much you're left with once you've minus the losses. So we use this in like net primary productivity for plants in year 13. If you're into economics, you talk about net and gross profits quite a lot, or like business studies as well. So yeah, useful word. How many reduced NADs have we produced? Oh, Khadija, you done it already? Yeah. 2 of these. And how many pyruvates? Yeah, 2 pyruvates as well. Cool. So how are we feeling with that? Good question. Is pyruvate the same as pyruvic acid? Honestly, better to Google it. I feel like maybe, but I'm not 100%. So we certainly would just talk about pyruvate normally for this. Lots of these molecules do have, a bit annoyingly, they have lots of different names for the same thing. So yeah, quite possibly it is. It is the same. Okay, nice. Yeah, lactate and lactic acid, again, yeah, another one with multiple names. And there's lots of these things throughout biology. How is hexose bisphosphate split into two? This one actually doesn't need an enzyme because it's so unstable. The phosphorylated glucose is so unstable, it pretty much straight away just spontaneously splits into two. That was the point of the phosphorylation of it. So yeah, the other stages, there are actually enzymes involved in lots of these stages. We don't need to know anything about them. The only ones we need are, we need Rubisco in the in photosynthesis, and I think that might be the only one. Oh, and ATP synthase, obviously, we need as well. All the rest, we don't worry about. So, yeah, this is quite a typical sort of question you'll get for respiration. They quite like drawing out the pathways, and then they ask you questions based on the diagram. So these can be a bit confusing because sometimes they put more detail than you'll have learned. Sometimes they put less detail than you'll have learned, and it's a little, you just have to orientate yourself with what's going on. Creatine is a short-term store of phosphates. It provides some extra ATP. It can give its phosphate to ADP. It can directly phosphorylate ADP when you need it. So yeah, it is related to this. There shouldn't be, I don't think there's anything else for ACI you need for this. No, this is kind of, it's quite standard between all the specs for this bit. So what is this representing? Firstly, yeah, nice, glycolysis. Very good. So, and where does this take place in the cell? Actually, we already had this question, didn't we? So hopefully this should be quite easy. Yeah, I would go for cytoplasm rather than cytosol. Just because that's definitely always in the mark scheme. Cytosol, don't know if you'd necessarily get that. So yeah, probably safer to go with cytoplasm. And then yeah, we need to identify D, E, and F from this diagram. So if you've got them, you can shout them out. D, ATP, nice. E is NAD. Oh, it does that sometimes. NAD. And F is, of course, pyruvate. Our end product. And so you can see here, this is a little bit different to what we looked at. So rather than we represented this first stage as just one step. So it's phosphorylated twice. In this question, they've actually split those up. So it first goes from glucose to hexose phosphate. That's just got one phosphate ion on it. And then you add another phosphate by using another ATP, phosphorylate it again. Then you get the hexose bisphosphate. So yeah, there are different ways of sort of showing this on the diagram. So what should we do? Probably got a little bit of time to do all these. Yeah, let's do this one. So if we want anaerobic respiration to keep working, we need certain things. So unlike aerobic respiration, if you need aerobic respiration to keep on working, you need enough oxygen. If you want anaerobic respiration to keep on working, you need enough NAD and you need enough glucose. So as long as you've got glucose and you've got NAD, you can keep doing anaerobic respiration. The problem is doing a lot of glycolysis. This is going to use up the NAD that's in the cell. The cell is going to run out of NAD at some point. So if you want glycolysis to continue, so this is glycolysis up here, this first bit. This is what we just looked at. Glucose to pyruvate. This makes our net, I can't write, our net 2 ATP. So this is good. We can make ATP. But eventually we're going to run out of NAD. So we can't keep doing this. So we need to regenerate the NAD. And this is where the anaerobic respiration comes in. So we made, this is our reduced NAD. And this is our oxidized NAD. So if we want to turn the reduced NAD back into oxidized, what we can do is we just take pyruvate and turn it into lactate. So essentially the pyruvate is becoming reduced and the NAD is becoming oxidized. Oh, sorry, the reduced NAD is becoming reoxidized back to NAD. So yeah, that's the main idea. Let me just check any questions. Why do they sometimes put acetyl-CoA after pyruvate? Yeah, this is because acetyl-CoA is involved in the link reaction, which is the next step if you're doing aerobic respiration. But we're not looking at aerobic today, but that would be involved with that. Glucose phosphate become isomerized into fructose 1,6-phosphate. Possibly, yes. But yeah, that's more than you'd need to know for A level. You can just talk about it as like a phosphorylated hexose sugar. The actual structure isn't relevant. But yeah, it could that does sort of ring a bell. For university, I had to learn all the stages. Sorry, got some sort of cough. I had to learn all the stages for respiration, and there's a lot more that you learn for A level. It's like something like 40 or 50 different steps and all the enzymes. And I crammed it, so I knew it at the time, and I knew it for my exams. But I like even a month after my exams, I couldn't draw it out from scratch anymore. So that's a problem with cram learning. I wouldn't recommend it. It works in the short term, but it doesn't stick in your brain for a long time afterwards. This is another redox reaction. So the reduced NAD is being oxidized, therefore pyruvate is being reduced. So lactate, this is just reduced pyruvate. And pyruvate is just oxidized lactate. So if you're doing this, you're creating a lot of lactate. This is the lactic acid that is building up in your muscles. When you finish doing exercise, so you finish doing anaerobic respiration, you're going to reoxidize that lactate back into pyruvate. So you're doing the reverse of this. That requires oxygen. That's why you need to keep breathing after you finish exercising. So what notes are we adding? In the absence of oxygen, no link /grabs /ETC. What else? So only glycolysis makes ATP. ATP, and it doesn't actually make that much, so it's not that efficient. To allow it to keep going, we need to have more NAD. So build up of NAD. So NADH is oxidize back into NAD, while pyruvate is reduced into lactate. So that's why we do, that's why we make lactic acid. That's why we make lactate. It allows glycolysis to continue. Therefore, we can make more ATP. So yeah, really helpful for the cell to make a little bit more energy in times where the aerobic respiration is not meeting the energy requirements. So that could be because the cells are located somewhere where there isn't much oxygen, maybe in a part of the body where they're sort of being starved of oxygen, or particularly in the muscles, if you're doing intense exercise, there's going to be not enough oxygen to meet the energy demands. So this allows some extra ATP to be created, useful for sort of like short-term bursts of energy. Cool. Then I think we haven't got time to go through the, well, I'll just very quickly write them out. So in yeast and plants, in fact, no, I think they're actually on, don't need to write them out because they're on here. Oh, well, they're sort of on here. So basically the equation is just saying in pyruvate gets turned into lactate in mammals, and pyruvate gets turned into ethanol and CO2 in yeast. And we sort of talked about this. This is used to make alcohol in brewing, and this is used to make bread rise. Makes fluffy bread, basically. So we use those processes for us, and this allows us to sprint bursts of energies. So that's kind of some of the benefits of these. Okay, so there's a few more questions. Fortunately, we didn't quite have time to go through everything. Yeah, sometimes we get through a little bit more. We do these classes on our normal website. If you subscribe to our tutoring packages, we do 5 of these classes a week. We only do 1 a month on YouTube. So yeah, if you wanted to do more, think about signing up. And yeah, I was going to talk a little bit more about the crash courses at the end. So yeah, you could think of ethanol being reduced pyruvate. It's not quite just reduced pyruvate because it's also lost a carbon. So it's like reduced pyruvate, and it's also decarboxylated pyruvate. So you could think of it like that. Yeah, lactate buildup leads to muscle cramp. Exactly. These sessions they're always at 5 for biology. I think other subjects might be at different times. But yeah, biology is at 5. So yeah, let's go through what these courses are all about. So like I said, we start with exam technique. So this is breaking down the questions and how you interpret it, and a lot of focus on the command words. For example, this one, we're looking at a suggest question. These are ones, they're actually like some of the hardest ones to answer in an exam because it's not something you'll have learnt about. It's where you've got to apply your biological knowledge and understanding in an unseen context, and then sort of give reasoned answers. So usually there's a lot of things you can say that score a mark. You just have to use your best judgment and what you've learned in an unknown situation. So they are tricky, and you can't really revise for them that much, but you can practice. You can practice doing them, but you can't pre-learn what you can say. So we'll go through all sorts of different questions. Make sure, I mean, hopefully you guys already know, but like, what's the difference between describe and explain? What do they want you to do? If they say compare and contrast, what does evaluate mean? All these things, you need to know them like really well. If you don't, this will be something that will really benefit your mark in an exam. And then we'll look at how you write sort of yeah, longer answer questions and how to structure your answers. These courses, it's a full day. So we start, I'm not sure exactly what time, maybe like 9 o'clock, sort of like 9 till 5, kind of like a longish school day. Yeah. So then we'll go through topics that people always find difficult, people always struggle with. So yeah, this is like looking at photosynthesis. And yeah, we'll do other ones. We'll probably do some stuff on inheritance, because I think people tend to like inheritance. It's quite an interesting topic, but it's also some of the hardest questions that you can do. So we'll recap inheritance. What else would we probably do? Maybe we do some stuff on muscles. That's quite a tricky one. Oh, nice. 10 o'clock till 5:30. Okay. Oh, you get a bit of a lie-in. So even better than a 9 o'clock start. Yeah, the chemistry will have their own topics that they're doing. So yeah, all the information is on our website. So I'll show you the website as well a little bit later. And you can actually start practicing what we learned in the first session on our exam technique. You can start practicing them straight away in this next section where we're looking at these difficult questions. Then these are the seminar booklets you get, sort of like biology Bibles, basically. They're going to really help your exam technique. So you can take these home. Yeah, filled with loads of extra questions, little tests, sort of all sorts of plenaries to continually test your knowledge. So it's going to help you do active revision, which is one of the best ways to make sure that you're improving. Loads of people don't actively revise. They kind of just passively read the book, or I don't know, half-heartedly make notes. It doesn't really help. You need to be really actively revising, continually testing yourself, and then retesting, and then doing that again a few days afterwards to make sure it's properly in, drawing connections, linking topics together. This is active revision. This will actually improve your grade and help you learn the topic. Passive revision, kind of a waste of time. It just makes you feel good that you're actually putting time into revision, but it's not going to really improve much. Anyway, this is the messenger thing. So you can message questions throughout the day, as many questions as you want, and people will answer pretty much instantaneously. So this is, I wonder if we could ever set this up as an actual service that we run year round, because it'd be pretty cool. I don't know if there's any possibility in that, but certainly during the courses, you can have your questions answered straight away. So yeah, pretty handy. These are the dates. So biology is the last one, 13th and 14th of April. Chemistry is that same week. Physics and maths are the week before. And yeah, Regent's University, really nice location. Or if you don't live nearby, you can join via live stream. This is where you book. So this is our website, snapprovise.co.uk. This is the seminars. And let me show you. I think I've got the website open. Hopefully. Yeah. So this is our website, snapprovise. And this, where is it? Seminars. So you go to product, you go to seminars, and then there's a bunch more information here on what we're doing. And you can book them directly on our website. Here are the other teachers. Some of you guys might have been to the YouTube sessions with Max, Matt, or Alyssa before. Yeah. And these are the package deals. So let me just remind myself what they are. Oh yeah, so buy 3, get 1 free. Everyone loves a sale. And what are these? Buy 4, 5, or 6 and get 2 free. So this would be, you'd have to be probably buying these with a friend. You're probably not going to go to 6. Well, I guess you could go to 6 seminars by yourself, because you could do the AS and A2. So actually, no, you could do it all by yourself, or you could combine with another person. Yeah. When is the deadline for paying for access? That is a good question. I presume there's no deadline for live stream, because the live stream won't be limited on places because we can do live stream for as many people as you want to attend. Yeah, basically when the seats sell out for in person. So yeah, I wouldn't, if you really want to come, I wouldn't leave it. I would probably just book it as soon as possible. Yeah. So yeah, first come, first serve basis, essentially. And the price is, it's 89 for a live stream, 99 for in person. Sorry, I think I said 79 for the live stream ones. Nice. So any questions on, yeah, good. Glad to hear that, Mohammed. And yeah, hopefully see you guys all in the seminar sessions. Any questions on the seminars or on the topic we went through today? I'll hang around for another couple of minutes to answer any questions. Otherwise, yeah, thanks for joining. And yeah, hope to see you in one of these sessions in the future and in our seminars at Easter. No worries. You're very welcome. Bye. Bye-bye. And yeah, I'll answer these questions. Carbohydrate lactate. Yes. It can be both transported to the liver or converted in the muscles. So yeah, it can be both places. The number for messenger will presumably be given out on the day of the courses. So yeah, I think that's not up and running yet. Any other questions? Okay, perfect. All right. Have a good evening, guys. Enjoy the rest of your Sunday and see you next time. Bye. Bye-bye.